Hello, everyone. Thank you for joining our School of Science the live event. And here we will have a life science talk about the popular science talk for life science. So I would like to pass the mic to our professor, life science professor, Professor Angela Wu, and she's going to give you a popular science talk on life science. Angela, please.
Okay. Hi, everyone. Hopefully you can see and hear me also. It's very nice to have you here today. So I'm the representative for Division of Life Science, and I'll start sharing my slides now. Okay, so as you can see, my talk today will be on reading and writing the genetic code and how that relates to diagnostics, GMOs, and designer babies. And this will touch upon a lot of cutting edge technology. So hopefully you will learn something new today, and also hopefully it will be fun. So first of all, again, welcome to HKUST Info Day 2021. If you have been to our campus, it's very nice. If you haven't visited yet, please come soon to visit us. Before I begin the technical part, I just want to introduce myself a little bit. I was born in Xi'an in mainland China. It's the oldest city in China and where the terracotta warriors are. And then after I was 5 years old, I went to Sydney, Australia to live with my parents, and I went to primary school in Sydney. And then after my primary school, that's when I moved to Hong Kong with my parents. And I went to secondary school in Hong Kong. I went to King George V. I don't know if there's maybe some of you in the audience who are familiar with that school. So after I graduated secondary school, I went to the United States for both my undergraduate education, also my PhD, and also my postdoc. So my undergraduate was at UC Berkeley, and then I did my PhD and postdoc at Stanford University, which is, they're all in California. I had a great time there. But after that, I decided to come back to Hong Kong and become a professor. So I joined HKUST in 2015, and this is my 6th year at HKUST. Okay, so to start my talk, I will just give you a brief introduction to what we call the central dogma of biology. Some of you may have taken biology before and may already be familiar with this, but as a quick reminder, the sort of fundamental blueprint of biology starts with the genome or the DNA, and that genome is then converted into RNA via the transcription of genes, and then those genes are made into proteins, and proteins are really the functional part that makes your cells function, and then your cells makes your whole body function. And so a simple analogy we can use is that we can view the genome as like a cookbook, right, of recipes where we can make lots and lots of different delicious food from the cookbook. But the cookbook itself is just the instructions, okay? You can't eat the cookbook, right? So the first thing that you have to do is take a recipe from the cookbook, and that's the equivalent of a gene. So a gene is one single recipe that makes one functional or edible item. So in this case, our protein then is the final cake that we have made from this one recipe. So that's kind of the relationship between a genome, a gene, and a protein, okay? Now, the first part of my talk, I'm going to talk about reading this genome, okay? Our genome is really, really long. Actually, it's made up of different letters, A, T, C, G. You may have learned about this in your class as well. So the genome is made up of a series of combinations of these A, T, and C, and G. And in total, there are 3 billion of these letters joined together to make our entire genome. So that's a lot of zeros and a lot of letters, right? So how did we know? I mean, how did we as people find out what our genome code is actually, what the order of those letters is, right? We want to find out, what is the A, T, C, and Gs that make up our genome, and also make up the different genes that make the different proteins. Because once we understand that, we can understand a lot about our biology and why we get sick, and et cetera, et cetera. So actually, recently, the next generation sequencing machine was invented, and this machine puts small pieces of DNA on the surface of a glass slide, and this technology will allow you to read all these short fragments of DNA at the same time. So we can read an entire human genome in one day. So today, with this next generation sequencing technology, I can take a sample of your saliva, and I can extract your genome from your saliva because your saliva contains some cells from your mouth. And once I extract that DNA, I can sequence it on this machine within 1 day, and it will give me all the 3 billion letters of your genome. So as I told you just now, because of this new technology that we have called sequencing, we can now read the genetic code. So what are some of the things that we can do now that we can read the genetic code? Oh, can I ask if everyone can, if people can see my slides? Can you see my slides? I got a chat message that I cannot see the slides. No? Oh, that's unfortunate. Okay, let me try again to share the slides. How about now? Okay, good. I'm sorry about that. Okay, let me just go back quickly then. So as I mentioned, the genome and the gene and the protein have these relationships to each other. Hopefully my verbal explanation, even without the picture, was okay. But if you have questions about this part, we can always come back to it later. And let me just repeat that our genome is very, very long, and we now have this machine. This is the machine that allows us to sequence our genome very easily. Okay, so then my question for you is, now that we can read the genetic code, what can we do with that information, right? So you can think about it a little bit yourself. But one of the first things that we can do is perform diagnosis, okay? Diagnosing certain diseases that are related to changes in our genome. So a simple example is Down syndrome, okay? Down syndrome is a syndrome caused by having an extra copy of chromosome 21. This chromosome 21 is just a part of the genome. So you can see all the rest of this person's genome is the same as a healthy person, but the chromosome 21 has an extra copy right here. And so what happens when you have extra chromosome 21 is in Down syndrome, these individuals are more easy to get infections. They're also more likely to get heart defects, and they're also more likely to get blood cancer at a younger age. They'll often be born with hearing and vision issues or develop them later. Plus, they have a higher chance and also earlier onset of Alzheimer's disease. So these are all very problematic, as you can imagine. And being able to diagnose Down syndrome means that if the parents can be prepared right in advance for a child with such issues, then they can have better preparation for this kind of issues. And also we can understand better the disease. So previously, before the existence of this next generation sequencing technology, the way that people have diagnosed Down syndrome is they have to insert this giant needle into the belly of the mother who is pregnant and suck out some of the amniotic fluid, which is around the baby. It's only in this way that they can get access to the baby's cells and then take the baby's cells and see lots of those cells to see whether it has an extra chromosome or not. But now that we have, I don't know about you, but this is pretty scary for me, and I'm pretty sure it's very scary for a lot of mothers. And actually this procedure does have a risk of causing miscarriage in pregnant women. So it's really not ideal. So now, today, what we know is that in the mother's blood, actually there's floating around very small fragments of DNA that comes from the baby, okay, because the mother's blood and the baby's blood is connected. So because this next generation sequencing technology is so powerful and so fast and much cheaper, what we can do then is just take a sample of the mother's blood from her arm, like a normal blood retrieval process, and sequence all the DNA in the mother's blood. Then just by counting the number of pieces of DNA that comes from the different chromosomes, we can evaluate whether there's an extra copy of chromosome 21 coming from the baby. So in this way, we have now a non-invasive prenatal diagnostic. Okay, this is non-invasive because it doesn't really, injure. It doesn't have a risk of injuring the baby nor the mom, because the blood taking procedure is very simple. You have all probably done it before. So this has actually saved many lives already, and this technology is now available in most clinics in many developed countries. And even if you ask around, if you have a friend or your parents' friends who might be pregnant or have been pregnant, if you ask them, there are some people who have already taken advantage of this new technology. So that's really great. I just wanted to give you one example of a cool thing that we can now do, because we can read the genetic code really quickly and really easily. But reading it is one thing, right? But can we write the genetic code? Can we change it? If there's, for example, for, chromosome 21 amplification, there's extra copy. Is there a way that we can edit the genome such that we can fix diseases even after the person is born? So the answer is we now do have new technologies that potentially can make these writing the genetic code very easy. So the first thing I want to talk about is that actually editing the genome, which you have heard about in the news lately maybe, editing the genome is not a brand new concept. Actually, GMOs, genetically modified organisms, have been around for many decades. And they are the result of a form of genome editing. Like, for example, a lot of the crops or fruits and vegetables that we eat, they are genetically modified. And even without the modern version of genetically modified vegetables, a long time ago, farmers were doing selective breeding, meaning that they cross plants with desirable properties with each other to breed a second generation with good features that they like, right? So this is already considered changing the genome of that crop. But once we have genetic tools, we started to do genetic editing directly on the cells of the plant, of these vegetables and fruits, to give us vegetables that have stronger growing power, or if they're resistant to flood or pests, all of which is really helpful to address issues around the world like famine. Now, this is what I call a more traditional form of genetic modification. But the issue is that this kind of genetic modification takes a long time to do, and it's more difficult. And later I'll show you that today we have more modern technology which makes this much easier. However, I also want to mention that there are lots of ethical issues around genome editing, including genetically modified foods, which you have probably heard about before. People might say, genetically modified foods are really bad because they affect your health. Actually, that part is not true. We have not seen any evidence that genetically modified foods will affect your health, but it can affect the ecosystem if we're not careful. So for example, if we make a genetically modified fish, which is bigger and stronger than the wild fish, and somehow this fish accidentally gets into the wild, it may outcompete all the weaker fish that are quote unquote natural and then change the ecosystem in a way that we do not want. So scientists are very careful to make this kind of GMO fish, make them not able to reproduce in the wild, so to prevent this from happening. But there are lots of advantages of the GMO foods. For example, as I mentioned, it can address issues of famine in the world. Like, for example, the starving children in some developing countries, they don't have enough food. So who are we to tell them that we should not make GMO foods and we should just let them starve? So there are pros and cons to this technology. And, this is something that we should consider both as scientists or just as, citizens of the world to think about and critically and think about whether we agree or we disagree with GMOs and for what reasons. So moving on to human health, how can genome editing help with addressing problems in human health? So one particular disease that is easy to illustrate this problem is thalassemia. So thalassemia is a form of anemia. What happens in thalassemia is that there's a mutation in the gene that makes red blood cells, okay? And with this mutated gene, the red blood cell is no longer round like this. It becomes this strange sickle-shaped, it's a strange shape, and its effectiveness to carry oxygen becomes much worse. So these people will suffer from anemia, okay? That's the reason why thalassemia patients suffer from anemia. Now, you can see from this picture, this is a picture of the gene, okay? And if there's a mutation in this location of the gene, then it will form the strangely shaped red blood cell because the hemoglobin protein will be not formed correctly. So if we can edit or change or modify this mutation in the sick person's genes back into the normal form, then presumably we can fix this disease for good and the person with thalassemia will be cured. So today we really do have a hope of actually fixing this using a technology called Crispr. You might have heard about Crispr in the news. It has been in the news a lot. And the way that Crispr works is that it has two components. The first component is a protein called Cas9. This protein actually comes from a bacteria. And there is another component called a guide RNA. So this is a small molecule which acts like a GPS for the Cas9. The function of the Cas9 is like a pair of scissors. And this small RNA is like the GPS. When you put these two things together inside the cell, this GPS will guide this pair of scissors to the right location on the DNA that we want to fix or edit, and the scissors will cut that place in the DNA right here, as you can see. So if we can cut the place where there's a mistake, then we can repair it using similar biological tools and potentially cure the person of disease. So this is the way that this new CRISPR-Cas technology works. Okay, so now that I've told you that it is possible to edit the genome of plants and crops, it's also possible now to use this CRISPR-Cas9 system to edit the genome of animals and even humans. Again, there may be lots of ethical issues involved in this genome editing, right? You can have a think about whether you think it is ethically acceptable to use genome editing on different types of species. But very quickly, let's have a discussion about the different types of genome editing. So I mentioned that we can edit the genome of different organisms, and we can classify them mostly into two categories that we care about, okay? We care about editing in non-human species, and we care about editing in humans or human tissues or human organs. So in the non-human species, actually, lots of this has been demonstrated in research labs and also sometimes already used in the commercial setting. So for example, in research labs, we have done genome editing on monkeys and mice to study health and disease in these model animals. We have also edited microbes like E. coli or yeast for production. For example, editing yeast to become a more effective producer of chemicals that we need, like drugs, and editing E. coli so that they can make better antibiotics for us. This is already in use in industry to make antibiotics and also a lot of food products. If you have heard of Impossible Meats in Hong Kong, they sell those meatless burgers, right? Those meatless burgers, actually the blood that you taste when you eat that meatless burger was made by bacteria and not an animal at all. There's also edited plants and animals that may be released to the biosphere, like crops, as I mentioned already, and also something called gene drive, which helps to reduce the incidence of malaria and other diseases carried by mosquitoes. So you can see that actually non-human genome editing is going on already, and it's very close to our lives. So the ethical issues of these, however, haven't been discussed as much. And I think it's because we as humans, we usually just think about ourselves first. So a lot of people, when they think about genome editing of humans, immediately there is some alarm and there's some fear maybe. But when we talk about non-human species, relatively speaking, the level of concern is lower. And so there has been less discussion in the public about editing of non-human species. But I would actually like to argue that we should participate more in the discussion of editing of non-human species because the advance is so fast and it's entering our lives already. We want to be prepared for any potential issues before that it happens rather than waiting until it happens to address them. Now let's go to human editing. So for human genome editing, we can actually split this category into two further subcategories, okay? One is the question of, is the genome editing happening outside the body or is it happening inside the human body? You can imagine that if the editing happens outside the body, we can control it better because it's in a dish, it's in a lab, and as scientists, we can do lots of things to check and make sure things don't go wrong. But if we directly do the editing inside the human body, then it's kind of difficult to control, right? We cannot just dissect the person to see if it's going well or it's going wrong. So this will become a lot more difficult to do, technically speaking. There's also a difference between what we call germline cell editing and somatic cell editing. And germline cells are essentially just the eggs and the sperm of the woman or the man. The reason why these two types of editing are different is because, oh, I kind of skip a few slides, is because the germline cells are the only cells that will pass down your DNA to the next generation. So if we make any kind of genome editing to the DNA inside the egg or the sperm, then also the children of that edited person will have those same edits or have a potential to have the same edits. But for the rest of the cells in our body, for example, our skin cells or our liver cells or intestines, even if we make some changes to their DNA, it will not be passed to the next generation. The edit will die with the individual that is edited. So this is the main difference, that the genome editing may be heritable or it may be non-heritable. And I think this concerns a lot of people when they think about genome editing. People immediately think about the heritable type. They think, oh, we're just going to change the entire mankind forever because we're changing the genome, we're going to make designer babies. Actually, that's not the case. Today, most of the time when we are talking about genome editing, we are talking about non-heritable genome editing. We are talking about maybe editing, for example, the red blood cell to cure a blood disease in a specific individual patient. And that edited change will not be passed to the next generation. So as you can imagine, the ethical issues of heritable versus non-heritable human editing will be very different. So in the real world today, there are lots of clinical trials going on to use genome editing to cure many, many types of cancer using a technique called CAR-T. So I've run out of time and I cannot talk about CAR-T in detail. If you're interested, you can read more about it later or ask me a question later. But even though there are still some risks of the genome editing going wrong, but for these patients who already have cancer and they may die, this is their only hope. So for them, the benefit of trying genome therapy will be greater than the potential risk coming from genome therapy. So the ethics of genome therapy, genome editing, really we should think about weighing the benefit of this therapy versus the potential risk and the harm. Like I mentioned, for cancer patients, their risk of dying from cancer is much higher than the risk of genome editing therapy going wrong. So for most cancer patients, they will really accept that genome editing is a beneficial thing. And that's why there is clinical trials allowed for this purpose. As for the germline editing of editing eggs and sperm, we are not sure if it will ever become mainstream because there's a question of whether do we really need it and are we really using it to treat disease or are we trying to do enhancement? This is where a lot of ethical issues will come in and people are still having lots of discussions about that. As you can see, the acceptance of gene editing is greatly higher for somatic therapy, meaning the normal non-egg or sperm cells, than for the germline therapies. Okay, so in summary, I just want to recap that we have talked about different technologies for sequencing the DNA and also the potential applications of those sequencing methodologies. And also, I briefly told you about the new genome editing technology called CRISPR-Cas and the potential application that it may have both for non-humans and for humans. Obviously, there are lots of ethical issues involved, so we really need to have a lot of discussion throughout the society with different roles in society participating in this discussion, including you guys as students, and also in the future, if you will become scientists, it's very important to think about the potential impact of your work on society. Okay, so that's the end of my talk. I hope that it has stimulated some thought about your opinion about genome editing and whether it should be acceptable. Thank you for being here today. Again, welcome to HKUST's Info Day. I'm happy to take any questions right now, if we have time.
We will have some, maybe 2 minutes. If there is any questions, you can just unmute yourself to ask the question or just type in the chat box. See if you have any question for the talk and also for our life science program. If no, then I will thanks again, Angela, to give a fantastic talk to us. And if you have any question about the life science program or also the admission for the School of Science, you can just go into the chat room under the Info Day platform. Now we still have some students and also the graduate in the in different groups like the biotech. I will have 3 programs, biotech and also a BISC and also the biochemistry and cell biology program. There, you can ask the question there. And thank you very much for joining us. And thank you, Angela, again. Okay.
Thank you. Bye-bye.
Bye-bye.